In this video, we're going to look at the structure of DNA in a little bit more detail, with emphasis on the nucleotide bases. But as a recap of my previous video, where I introduced to you some key terms that you need to know for your GCSE. The first of which is the genome, which is the entire genetic makeup of the organism, and you can find the genome in the nucleus of every cell. The genome is in the form of chromosomes, of which in humans each cell contains 46 chromosomes or 23 pairs, apart from the sex cells. The chromosomes contain linear sequences of genes, so genes that are coding for the proteins inside the cell, proteins such as enzymes or hormones. The genes, or the chromosomes, take the structure of DNA, or made of DNA. The DNA is this large polymer twisted into the double helix. It's represented by the picture on the right hand side. The information contained within DNA, or in other words genetic information, is due to the sequence of nucleotide bases that run through the DNA molecule, and you can see that they are represented by those coloured bars in the centre of the DNA, the blue, yellow, red and green. They represent adenine, or the letter A. There's thymine, T, cytosine, C, guanine, G. Now let's have a look at the structure of nucleotides in a little bit more detail. Let's just focus on one of them. It's a little bit more than just a base. Each base, whether it's adenine, thymine, cytosine or guanine, each base is attached to a simple sugar, which is also attached to a phosphate. And that's every single base, whether it's adenine, thymine, guanine or cytosine. In terms of where the sugar and phosphate are, they're actually running along the strands of DNA, which are represented by the blue spiral and the red spiral. So all the way along, every time there's a base, there's also a sugar and a phosphate. But let's focus a little bit more on the base now. If you have a look at the DNA molecule on the right hand side, adenine is represented by the yellow bases. Which base is it always paired with? Adenine, yellow, is always paired with a green base, which represents T, every single time. If you look at cytosine, which are the blue bases here, they're always paired with guanine, the red bases, every single time. So, to summarize that, the nucleotide base adenine always pairs with thymine, thymine always pairs with adenine, guanine always pairs with cytosine, cytosine always pairs with guanine. A to T, G to C. The technical term for this is complementary base pairing. A complements T, G complements C. Complementary base pairing. This is useful for when we try to understand how DNA copies itself, and it has to. Every time a new cell is made, the DNA has to be copied so that new cell has the DNA. So let's take a closer look at how useful this is. If we focus in on the blue strand of the DNA, if we go from the top, the blue base there, cytosine, is followed by the one underneath it, yellow base, adenine. Apologies for the cytosine being upside down. That's not intentional and doesn't affect us in any way, but it's still cytosine. So cytosine followed by adenine. Adenine is followed by guanine. And guanine is followed by thymine. Now, because of the complementary base pairing, the next sequence of bases on the other strand has to follow the rule that cytosine pairs with guanine and adenine always pairs with thymine. So let's fill in the missing nucleotide bases on the red DNA strand. Cytosine pairs with guanine. Adenine pairs with thymine. The guanine will now pair with cytosine. And finally, thymine will complementary pair with adenine. This rule will repeat itself all the way along the DNA molecule. So using that principle of complementary base pairing, write down what the sequence of nucleotide bases will be in the new DNA strand that will complementary base pair with a sequence of bases C, G, A, C, 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 G, A.